Hello everyone, a very good evening. Am I audible? Please reply if I'm audible. Please let me know if I'm audible. Okay, so on behalf of Intelletics, I welcome you all for this session wherein I'm going to teach you the 10th standard ICSE students the chapter that is cell division, how the new cells come into existence from the pre-existing cells. As on 15th, you all have got your bio exams. So this, on behalf of Intelletics, this is just an attempt to help you out by reading word to word and explaining every term and what is important to you, especially for the exams. So please stay connected and we shall start. And as we understand one important thing, that is there is a big lag after what I mean when you receive my video and the moment I'm talking, there's a big lag. And for this reason, I shall tell you, I would tell you to, or rather, I would request you only one thing, that is, please, if you have your doubts, any difficulties, we please type out a message. Okay, if it reaches me on time, certainly within the lecture, I shall talk about it. I shall clarify your doubts. But if it doesn't, in that case, I shall definitely answer your queries in the next session whenever we will be conducting up a live streaming. So, with this, I start with the chapter Cell Division, New Cells from the Existing Ones. Now, we know that cell is the basic unit of life. Whatever functions are performed by any organism, be it a plant, be it an animal, be it a unicellular organism, ultimately, these functions are performed by the cells itself. Every cell performs one or the other functions. And remember, every cell has its own lifetime. For example, red blood cells in our body have a lifetime of only and only 120 days. This point perhaps is not mentioned in your textbooks, so I would like to tell you this. Please remember, our RBCs have a lifetime of 120 days. Now, what happens is when the cell is about to die, or if the cell dies for any reason it might be, the functions performed by the cells will be stopped. And the body part, or the body organ, the functioning of that organ would be affected. But the body does not let this happen. Why? Because the cells reproduce, that is, the cells, they undergo division, they produce their own copies. And this is how the body ensures that the body functions are not affected. Now, the new cells need to be produced for four reasons. First, for growth. Second, for replacement of the old, dead, worn out cells, injured cells. Third, repair. As you know, the wear and tear of the body keeps going. So we need to repair up the cells. So for the repair purpose. And fourth is for reproduction. That is, one organism gives rise to another organism similar to itself, and that process is reproduction. So, MCQs, I can ask you cell division is essential for dash A. Growth and replacement, B. Repair, C. Reproduction, D. All of these. So, remember, it should be all of these. Now, coming to the first one, that is for growth. Every organism, be it a plant or an animal, begins its life as a single cell, that is the fertilized egg. Remember, friends, the fertilized egg, the first cell of our life, is called as zygote. So you and me, or all the higher organisms for that matter, begin their life from a single cell, which is called as zygote, or which is a fertilized egg. But are you and me unicellular? No, we aren't. This means that even though our life starts from a single cell, this cell, it keeps on dividing and redividing and redividing, and result is a huge organism can be produced. So this cell, the one which is formed as the first cell of a life, I mean the zygote or the fertilized egg, it divides repeatedly to form a cluster of cells which starts shaping for different functions to form tissues and organs, and thus cell division is essential for growth. Remember, different cells come together to form the tissues. Tissues come together to form organs. Organs come together to form organ systems. Organ systems come together to form an organism. So, first purpose for, or the first need, or the first scale of cell division is that for growth. All the living organisms, which I'm talking about multicellular organisms, they begin their life with a single cell, that's zygote or the fertilized egg, and which undergoes cell division repeatedly, and as a result, multicellular organisms are produced. Now, in the given image, you can see this is an image of a 4-day-old human embryo which contains only 16 cells. So, fourth day, the number of cells in the body, human embryo, is just 16. We have started our life journey from a single cell. So a single cell undergoes mitosis to form two cells. Then those two cells undergo mitosis to form four cells. Four cells undergo mitosis to form eight cells. And eight cells undergo mitosis to form 16 cells. So four mitotic divisions have been undertaken by the time we are 4-day-old as a human embryo, thereby helping us to be 16 cell strong. As they multiply, they specialize to form the tissues and organs of the body. Coming to the next point, for replacement, there is always a wear and tear of the cells during the normal body functions. For example, 20 million red blood cells in our body are destroyed every minute. Very, very important point. 20 million red blood cells in our body are destroyed every minute, and these are replaced by the new cells. So it's not that they're just destroyed, they're equally going to be replenished by those many number of cells every day, so that the number of cells in our body tends to remain constant. And these RBCs are produced in the bone marrow. Please remember this. In the adults, it's the bone marrow which helps in the production of the blood and the blood cells. What I mean to say is that RBCs, WBCs, platelets, all of these cells are mainly produced in the bone marrow of that of adults. I'm specifying here of that of adults. The reason is because in the embryonic stage or in the fetal stage, the new cells are produced in some other organs. Please remember, bone marrow is nothing but it's a tissue that is present in the hollow, that is the open hollow space inside the long bones of the body. In plants, the old and the dried leaves fall off and the new ones grow out. So for the new cells, the new leaves to be produced, cell division becomes essential. So this is how cell division helps for replacement. Coming to the next, for repair. Apart from normal wear and tear of the tissues in the body, there may be accidental injuries. One may get cuts in the skin or fractures in the bone. Repair of such injuries is again through the cells which divide, cover up the gaps and join the broken ends. Okay, so cells also perform the function of healing, repairing, in any injured area. Coming to the next point, in cell divisions for growth, replacement, and repair, the number of chromosomes remains the same at each division. The chromosomes duplicate and distribute equally in the daughter cells. This kind of division, which occurs in all the body cells except in the egg-producing oocytes in the ovaries and sperm-producing spermatocytes in the testes, is known as mitosis. I shall highlight this point. So, the cell division taking place in the body cells, all the body cells other than the oocytes and the spermatocytes, is known as mitosis. This, you have to understand that the cells which are present in the ovaries and in the testes, that is the oocytes and the spermatocytes, undergo a different kind of cell division which is called as meiosis. The same question can be asked to you in some different form. For example, I can ask you, mitosis takes place in dash. A. Somatic cells. B. Body cells. C. All the somatic cells in the body except the gonads or the reproductive organs. So your most perfect answer for this question will be C. Remember, our body is a collection of millions of cells. The cells that are present in all the body parts other than the reproductive organs are called as the somatic cells, which undergo mitosis. Whereas the cells which are present in the reproductive organs are called as the germ cells. Please remember germ cells. Germ cells are the cells that are present in the reproductive organs, that is testes in the males and ovaries in the females. And these help, or rather these undergo meiosis, and thereby they are involved in reproduction. So remember, oocytes in ovaries and spermatocytes in testes undergo meiosis. So friends, please remember, germ cells are the cells in the reproductive organs which undergo meiosis. Now, coming to the next point, number 4. Reproduction is also through the activity of the dividing cells. Amoeba or bacteria just divide to produce two similar independent cells by mitosis. In higher forms, as in humans or in the banyan tree, special cells in the reproductive organs undergo a special kind of cell division, that is meiosis, to produce sperms and eggs. The sperms and eggs receive only half the number of chromosomes of their parent cell, that is, one chromosome from each pair. This reduction in chromosome number is very significant. That is important. Keep this in mind. Okay. Remember, meiosis is the cell division which helps in the production of the chromosomes, that is, from diploid to haploid. The number of chromosomes present in any organism is fixed. For example, in human beings, all the human beings in the entire world, other than the exceptional cases who have some disorders, all the living organisms in the entire living world are made up of 46 chromosomes. Remember, each and every cell that we possess consists of 46 chromosomes. Okay. Only and only the sperms and the eggs produced by any animal will contain only half the number of chromosomes, that is 23 chromosomes. So this constancy in the number of chromosomes is also achieved because of a special kind of cell division that is called as meiosis. Now, coming to this tabular form, the body cells, that is the somatic cells in man, contain 46 chromosomes, as well as in women. There are 46 chromosomes. We call it as 23 pairs, and we write it as 2n, where an n represents the number of chromosomes So 2n is double set of chromosomes. Friends, please remember, whatever chromosomes that we have got it from our parents. So out of 46 chromosomes that everybody, each of us possesses, 23 chromosomes we have got it from our father and 23 chromosomes we have got it from our mother. So we say 23 pairs of chromosomes, that is 46 chromosomes, are present in our body. Coming to the next, that is the sex cells, sperms and the eggs. The sperms and the eggs contain only a single set of chromosomes. That is, the chromosomes in a sperm and the chromosomes in an egg are just 23. Whenever fertilization takes place, remember, in biology, fertilization means the fusion of the haploid male gamete with the haploid female gamete to form a diploid zygote. So after fertilization, or rather fertilization results in the formation of a diploid zygote. Imagine if 46 chromosomes, if a cell with 46 chromosomes fuses with another cell with 46 chromosomes and a new organism is born. In that case, what will happen is the new organism will have 46 plus 46, that is equal to 92 chromosomes. But does that happen? No. This means that the chromosomes or the cells that fuse with each other have only a single set of chromosomes. Hi Dibyan Sinha Pillai. Hi Phoenix. Hi Zara Sheikh. If you have any doubts, you can post them up. That's not a problem. Any doubt you have got? Now, coming to the next, that is types of cell division. There are two types of cell division, mitosis and meiosis. So mitosis is the cell division leading to growth and development, whereas meiosis is the cell division leading to the production of gametes, that is 6 cells. Now to start talking about mitosis. The word mitosis is being coined from two words, mitos, that is thread, and osis, that is stage. So referring to the chromatin thread stage. Mitosis is a cell division in which one parent cell divides into two identical daughter cells. Please remember the word used here is identical and not similar. I hope you know the difference between similar and identical. The basic difference between mitosis and meiosis. There are lots and lots of differences between mitosis and meiosis basically. Mitosis is equational division, whereas meiosis is reductional division. Mitosis occurs in the somatic cells, whereas meiosis occurs in the germ cells. In mitosis, one cell produces four daughter cells, whereas in mitosis, one cell produces two daughter cells, and in meiosis, one cell produces four daughter cells. So there is a big, big difference between mitosis and meiosis. Mitosis, the cells that are produced in mitosis are identical to the parent cell, whereas the daughter cells that are produced in meiosis, the daughter cells that are produced in meiosis are different from that of the parent cell. So mitosis is the cell division in which one parent cell divides into two identical daughter cells. The most important aspect of mitosis is that the same normal chromosome number is maintained at each cell division. Just before the division of the cell, the nucleus prepares for this change and doubles the quantity of DNA, that is the chromosome substance. This is the interphase. Remember, DNA quantity is doubled in the S phase of interphase. S phase stands for synthetic phase. So it is a synthetic phase in which the DNA quantity is doubled. And this is S phase of the interphase, which is apparently the resting phase, since no change in chromosomes is visible externally, but actually it is quite active in synthesizing the DNA. Very, very important point. Interphase is apparently called as a resting phase, since no change in the chromosomes is visible externally. I can ask you this as an assertion and reasoning type of question. Now, please remember earlier, interphase was called to be as a resting phase, but later on it was known that interphase, in fact, is the phase wherein the cell shows its highest metabolic activities because the cell prepares itself for cell division. Interphase in turn consists of three phases: the G1 phase, the S phase, and the G2 phase. So remember, S phase is that phase in which the DNA doubles up, that is, DNA forms its own copy, as well as it also results in the synthesis of histone proteins. Please remember, histone proteins are synthesized in the S phase, along with the formation of, or along with the duplication or replication of DNA. Now, in any cell division, it might be either mitosis or meiosis. The first thing that happens is the division of the nucleus. And the division of the nucleus is called as karyokinesis. The word karyokinesis comes from two words: karyon, that means nucleus, and kinesis means division. And this karyokinesis is followed by cytokinesis. A cytokinesis is the division of cytoplasm. Karyokinesis is the division of nucleus. Now let us talk about the phases of mitosis. So as I said, the first one is karyokinesis. Karyokinesis of mitosis occurs in four main phases, although each of these phases merges into the next phase, thereby making it a continuous process. The four phases of mitosis are prophase, metaphase, anaphase, telophase. Please remember, friends, you cannot change the sequence of the same. So first phase is prophase, second phase is metaphase, third phase is anaphase, and fourth phase is telophase. By and large, the four phases of mitosis are similar in both plant cells and animal cells. However, we are first describing the mitotic stages in an animal cell. And this figure 2.2, I shall show that, illustrates these changes in the animal and plant cells side by side. Two major differences in the mitosis of plant and animal cells have been listed in the tabular form for this 2.1. Let us have a look at it. These are the changes that have been shown. I shall talk about it at every step. Please remember, the plant cells and the animal cells differ mainly with respect to cell division. There are 2 points of differences we need to remember. The first thing is, the plant cells do not show the presence of centrioles, whereas the animal cells do show the presence of centrioles. Second point is, the plant cells during cytokinesis, that is the division of cytoplasm, they divide by means of a cell plate formation. Whereas in an animal cell, there is no cell plate formation, but there is just a constriction formed at the center of the cell. Now, let us start talking about prophase. The word pro means first. So let us first talk about the prophase of mitosis. The chromosomes have become short and thick and are clearly visible inside the nucleus. Please remember the chromosomes are visible very clearly, but the sister chromatids are not really very clear. Even though they are present, they are more, what you can say, distinct. They are more distinctly seen in the metaphase. Each chromosome has already duplicated, that is, it has made its copy to form chromatids. Remember, the chromatids of the single chromosome are called as sister chromatids, which are connected with each other by a centromere. The two sister chromatids remain attached to each other at a small region called as centromere. Coming to the next point, the centrosome. The centrosome is nothing but the area which holds the centrioles. Okay, so don't get confused between centrosome, centromere, centriole. Okay, so centrosome is nothing but that part of the cytoplasm which appears a bit denser because of the presence of the centrioles that's near the nucleus only in an animal cell. The centrosome in animal cells splits into two along with simultaneous duplication of the centrioles contained in it. So you normally find two cylindrical structures present at right angles to each other as one centriole. The daughter centrioles move apart, that is, they separate from each other and occupy opposite poles of the cell, that is, they go on two ends of the cell. Each centriole is surrounded by radiating rays, which is termed as aster. The word aster means star. Please remember, it's either called as aster or astral rays or astral fibers. And the reason why they are called as aster is because, like the stars, as the stars, they show their radiations. Okay, I mean, their what you can say, the light splits. In the similar way, these fibers appear to be splitting from a single source. And this is the reason why they are called as astral rays or astral fibers, or simply at times called as aster. The animal cells may have either one or two centrioles. So the number of asters is also one or two. A number of fibers appear between the two daughter centrioles, and these are called as spindle fibers. Friends, please remember they're called as spindle fibers because of the typical shape. If you observe the diagram later on, I would show why is it called a spindle. The reason is because these fibers, they are slightly broad at the center, but at the tips they are quite tapering. Such a shape is called a spindle shape. And because these fibers have a spindle shape, these fibers are called as the spindle fibers. And there are different types of spindle fibers as well. The nuclear membrane and the nucleolus disappear. This happens in the prophase. Remember this. The duplicated chromosomes start moving towards the equator, that is center of the cell. Please remember the poles mean the extremities of an axis and equator means the middle plane, that is the central area dividing the cell into two similar halves. The naturally occurring cells may be oriented in all possible directions. Now let's talk about metaphase. Before that, I shall show you the diagram. So here you can look at this diagram, the early prophase and the late prophase, so that you understand. Even please Look at this image carefully, zoom in and see. You will also find the difference in the diagrams of interphase, early prophase, and late prophase. In the interphase, you find the chromosomes are not seen. It is just seen as a network of threads, which we call it as chromatin network. Whereas in the early prophase, we see the chromosomes that are distinct. And in the late prophase, we also find that the centrioles have almost reached the poles. The nuclear envelope, as well as the nucleolus, it's on the verge of disappearing, and they are almost reached the poles, and also the formation of spindle fibers has started. So please see this diagram carefully. In the exams, you may be shown any of these diagrams. They may give you any of such kind of images, not the same one, but similar to this. And you may be asked to identify that stage. Then there will be many sub questions like identify which is the next stage, identify which was the stage before that, yeah, identify which is the typical feature of that stage, and so on. I hope you have seen this properly. If you have any doubts, you can certainly ask me. It's never a problem. So here we start with the prophase itself. So we had a talk about the prophase. Now we talk about metaphase. The word meta means after. So this is something which is after prophase. So it is called as metaphase. Meta means after. This can be asked to you, what is the meaning of the word meta? So the duplicated chromosomes arrange on the equatorial plane. Remember, equatorial plane is nothing but the center of the cell. Each chromosome gets attached to a spindle fiber by its centromere. So I can ask you an MCQ in this. I can ask you the spindle fibers connect to the dash. So remember, the spindle fibers are attached at the centromere region. Please remember this, the spindle fibers are attached at the centromere region, okay, of the chromosomes. So remember that the centromere not only helps in connecting the two chromatids with each other, but the centromere also helps in attachment of the spindle fibers. Now coming to the next, that is anaphase. Ana means up, that is back. Okay, the centromere attaching the two chromatids divides, and the two sister chromatids of each chromosome separate and are drawn apart towards the opposite poles. So please keep this in mind. The centromere, it divides in the anaphase, a very important point. So remember, in anaphase, the centromere divides. This is important. In case of metaphase, the duplicated chromosomes, they arrange themselves on the equatorial plane, and spindle fibers are completely formed. Okay, in anaphase, the centromere attaching the two chromatids, it divides, and the two sister chromatids of each chromosome separate and are drawn apart towards opposite poles. The drawing action is performed by the contraction of spindle fibers. Even this can be asked to you as an MCQ, that the centromere and the chromatids are drawn towards the poles due to dash. It is due to contraction of the spindle fibers. Coming to the next, that is telophase. The word telo means end. Each chromatid or the daughter chromosome lengthens, that is, it becomes long. It becomes thinner and turns into a network of chromatin threads. Friends, simple thing, please remember, The changes that happen in the telophase are exactly opposite to the changes that take place in the prophase. So if you remember, in prophase, chromatin network gets condensed to form chromosomes, and in telophase, exactly opposite thing happens. That is, chromosomes get converted into chromatin network. In the prophase, nuclear envelope disappears. In the telophase, it reappears. In the prophase, nucleolus disappears, and in the telophase, it reappears in each daughter nucleus. All the contents of the cytoplasm, including the mitochondria and also the chloroplasts in the plant cell, are randomly distributed in the daughter cells during karyokinesis. So this is all about telophase you need to remember. Now coming to the next, that is cytokinesis, that is division of cytoplasm. At the end of telophase, a furrow appears in the cell membrane in the middle. Furrow is nothing but a constriction. Please remember, so cytokinesis, it is about is formation of furrow. It occurs at the end of the telophase, not in the start of the telophase, but at the end of the telophase, the formation of furrow starts, and later on, this furrow deepens and divides the cell into two daughter cells in case of cytokinesis. As I've mentioned earlier, cytokinesis is the division of the cytoplasm, whereas karyokinesis is the division of the nucleus. Now, look on the screen, the interface. You can actually compare the diagrams. So there is no visible change in the chromosomes in the interface. Very, very important. But there is active synthesis of DNA. As I already mentioned, DNA is synthesized in the S phase of interphase. Here you can see prophase A and B, that is early prophase and the late prophase. Centrioles start moving apart and reach the opposite poles. Chromosomes become distinct. Chromosomes are already duplicated as paired chromatids. As I've told you, chromatids are formed in the S phase, but they are not distinctly seen in the S phase, but they are seen very clearly in the prophase and most clearly in the metaphase. In fact, if I ask you a question that if you want to study the structure of the chromosomes in a cell, which would be the best stage to study that? Well, the answer to this question is metaphase. So if you want to study the structure of the chromosomes perfectly, the ideal stage is metaphase. Keep this in mind. Sister chromatids attach to each other at a small region called a centromere. Spindle fibers appear between daughter centrioles. Nuclear membrane and nucleolus disappear in the prophase. So look at this particular image. If you have any doubts, please, 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 you type those doubts here. You can post it as a message. I will certainly get back to you if you have any doubts. The next one is metaphase. The metaphase, each chromosome gets attached to the spindle by its centromere. And remember, in metaphase, the chromosomes are present at the equatorial region, that is center of the cell. So you can have a close look, you can zoom in the screen and see metaphase. Now coming to the next one, that is anaphase Centromere attaching the two chromatids divides in the anaphase. As I have already mentioned, centromere divides in the anaphase. The two sister chromatids of each chromosome separate and are drawn apart towards opposite poles, pulled by shortening of the spindle fibers. So remember this, the shortening or contraction of spindle fibers results in the pulling action of the chromosomes towards the opposite poles. A furrow starts in the cell membrane at the middle in animal cell. So you can look at this image closely. So please look at it, and if you have any doubt, you can definitely ask me. Okay, so going ahead, a furrow starts in the cell membrane at the middle in the animal cell. Please do not forget this. Furrow is nothing but a constriction. So either I can ask you it as a constriction, yeah, I can ask you as a furrow. Okay, it means the same. Coming to the next, that is telophase. Two sets of daughter chromosomes reach the opposite poles. Okay, that is telophase. Spindle fibers disappear. Chromatids thin out in the form of chromatin fibers. Nuclear membrane is formed, and the cleavage furrow starts deepening in the animal cell. The nucleoli reappear. You can look at the image and you can understand it better. So it's very, very easy to remember. All the changes that do take place in the telophase are exactly opposite to that of the prophase. Coming to the next, that is cytokinesis. Cytokinesis, that is the division of the cytoplasm. The cleavage furrow, that is constriction, deepens totally in the animal cell and separates the two daughter cells. This is what happens in cytokinesis. Now, karyokinesis and cytokinesis. You have to remember all the nuclear changes that occur during cell division are collectively termed as karyokinesis. And all the changes that do happen in the cytoplasm are collectively called as cytokinesis. So karyokinesis is followed by the division of cytoplasm, that is called as cytokinesis. Let us understand the differences in mitosis in the animal and plant cells. The nuclear and chromosomal event of mitosis in plant cells and same are the same as those in animal cells, but there are some differences. First difference is asters are formed in an animal cell. They are not formed in the plant cell. The reason behind it is the asters are produced by the centrioles. Centrioles are present in animal cell but not in plant cell. So this also can be written as a point of difference. Second, cytokinesis in case of animal cell occurs by furrowing of cytoplasm. That is, it undergoes a constriction at the center. Whereas cytokinesis in the plant cell occurs by the cell plate formation. In case of animals, it occurs in most tissues throughout the body for growth and replacement. Whereas in case of plants, mitosis occurs mainly at the growing tips for lengthening and sides or increase in the girth. That is, in simple words, it occurs in the meristematic cells. Now, the terms parent and daughter cells. Both these terms are actually inappropriate for two reasons. One, the parent cell loses its identity in the process. And two, the daughter cells are not necessarily feminine. However, these two expressions are being used conventionally. So please remember this. They're not feminine cells. In fact, they do not have any gender as such. Okay, of course, the sex cells will decide, the gamete fusion will decide whether that's going to be a boy or a girl. Coming to the next, significance of mitosis. Significance is nothing but importance. The growth or increase in the body size due to formation of new cells in the tissues. Second, repair of damaged and wounded tissues by renewal of the lost cells. Third, replacement of the old and dead cells, such as the replacement of the blood cells and the epidermal cells of the skin. Fourth, asexual reproduction in which the unicellular organisms such as amoeba or the yeast cell divide into two. Fifth, it maintains the same chromosome number in the daughter cells. Now, the diagram here shows the sectional microscopic view of an animal tissue undergoing active mitotic division. Observe carefully that such naturally occurring cells are oriented in all possible directions and also they are in different phases. Note the stages of the cells numbered 1 to 6. One you can see is the interphase. Second, the cell in the prophase stage, advancing condensation is seen. Third, the cell is seen in the metaphase stage, that is, the chromosomes are seen in the equator. Fourth, the cell is seen in the anaphase stage because the chromosomes get pulled to the poles. Fifth, the cell is seen in the telophase stage, that is, chromosomes reach the poles and the nucleus is being formed. And six, young cells are formed after division. The mitochondria and chloroplasts in cell division. So any doubt with this? Before we start with any doubt in this? The mitochondria and the chloroplasts in the cell division. Both mitochondria and chloroplasts have their own DNA, that is, they have their own genes. They also contain their own ribosomes, which help in producing the particular proteins of these two organelles. Please remember, this is the reason why the mitochondria and the chloroplasts are called as semi-autonomous structures because they are in a way self-sufficient. So they are called as semi-autonomous structures. Both these divide on their own by simple fission just by splitting into two and a partition between the two daughter cells produced by mitosis. Very, very important point, friends. So they divide only by simple fission. I can ask you this. Okay, they divide by simple fission. Mitochondria as well as chloroplast. Mitochondrial division is also guided by the genes in the nucleus and through the cytoplasmic ribosome. Another important point. Now, how old are some of our body cells? Cells of the eyelids, nerve cells of the cerebral cortex, and the muscle cells, and most muscle cells last a life once dead are not replaced. Remember, the neurons and most of the muscle cells, they last for a lifetime. In other words, they do not divide. Skin cells are replaced every 2 weeks or so. Red blood cells last for about 120 days and are replaced. Bone cells are replaced every 10 years or so in the gut. Epithelial cells lining the gut last only for 5 days, and the average life of other gut cells is about 15 years. The liver cells are replaced every 300 to 500 days. So these are the important points that you need to remember. Now coming to the next, that is cell cycle. In fact, this has to be ideally done first, much before cell division is explained. Cell cycle, that is divide, grow, and redivide. The new cells at the end of mitosis are relatively small, with a full-size nucleus but relatively little cytoplasm. Please keep this in mind. The cells that are formed new is the daughter cells. When they just form, they're smaller in size, with smaller nucleus and smaller amount of cytoplasm. This can be asked to you in the MCQs. Now they enter the interphase, during which they prepare for the next cell division and grow to the same size as their mother cell. The interphase itself has 3 phases: the first growth phase, the synthesis phase, and the second growth phase. Please remember the sequence. This is also called G1, that is first growth phase, S, that is synthetic phase, and G2, that is the second growth phase. First growth phase, that is G1. In this phase, RNA proteins are synthesized. The volume of the cytoplasm increases. Now, why does the volume of the cytoplasm increase? Because everything that is present in the cell is growing. Mitochondria in all the cells and chloroplasts in the plant cells, they divide, and these two organelles have their own DNA. In late G1 phase, all cells must follow one of the two paths. They may either withdraw from the cell cycle and enter a resting phase, that is R, or start preparing for the next division by entering the next synthesis phase, that is S. Just for your additional information, I shall tell you that this resting phase, that is when the cells withdraw from the cell cycle, that phase is also called as the G0 phase. Synthesis phase. More DNA is synthesized and the chromosomes are duplicated. So remember, synthesis of DNA and duplication of chromosomes is seen in the synthetic phase. Then the second growth phase. This is a shorter growth phase in which RNA and proteins necessary for cell division continue to be synthesized. Now the cell is ready to start cell division, and thus the cell cycle goes on. Please remember, synthesis of RNA and proteins, it starts in the G1 phase, but it is completed in the G2 phase, that is the second growth phase. So you can have a close look at this diagram. You may be given this particular image, and the labelings, instead of these labelings, they may give it up as A, B, C, D, and you will have to identify which stages, what exactly does that A, B, C, D refer to. So please zoom in and see this image properly. Register it in your mind, because such image-based questions are certainly asked. Now, can the cell cycle go on endlessly? No. At some places, it stops permanently. This is important. The cell cycle at some places stops permanently, and at some places it stops temporarily, and others till it is lit. So there is a regulatory mechanism for the cell cycles. Brain and the other nerve cells, that is neurons, once formed in the embryo, They do not divide further. Once dead, they are not replaced. Liver cells, on the other hand, may divide only once every 1 or 2 years to replace damaged or destroyed cells. As we have already done this point, surface skin cells are continuously lost and replaced by the underlying cells. A large portion of household dust contains human skin cells. The powdery material that comes off from your skin on scratching or on hard rubbing while bathing contains the same dead cells. In plants, the cells at the growing points, that is meristems, divide very rapidly and produce new leaves, buds, and flowers. Now, this is important. The meristematic cells are the cells which are actively dividing cells in the plants that produces the new leaves, buds, and flowers. Now, remember, specialized germinal cells, which we can even just call them as germ cells, in the ovary and testes in the animals and in the ovary and anthers in the plants, undergo the other type of cell division called meiosis to produce sex cells. The sex cells are also at times called as gametes. Also remember, the uncontrolled non-stop cycles may lead to tumors and that may not be cancerous. Now let us talk about cell production and cell death. As children, we grow because new cells are continuously being produced and they outnumber the dying cells. When we are adults, our cell population stays constant. The number of new cells produced equals the number of cells dying. As we grow old, the number of new cells produced runs short of those that are dying. And this is the real reason why we say we are aging. So in case of aging, the rate of cell division, it goes down. So when we are adults, our cell population stays constant. The number of new cells produced equals the number of cells dying. Now coming to the next one, meiosis. Meiosis is called as reduction division. That is the one which results in the production of gametes. Gametes, as I already mentioned, are the sex cells, and please remember all gametes, be it in plants or be it in animals, are haploid. That is, all the gametes contain a single set of chromosomes. Meiosis is the kind of cell division that produces the sex cells or the gametes. It takes place in the reproductive organs like the testes and the ovaries in humans, and that produces sperms and ova. Okay, so sperms and ova are nothing but the sex cells, also called as the gametes, which produce as a result of meiosis. In the flowering plants, it takes place in the anthers, which you know is the male part of the flower, and ovary, which is the female reproductive flower part of the flower, and that produces pollen grains and ovules. The most significant aspect of meiosis, that is, meiosis means diminution, that is, reduction, is that the number of chromosomes in the sex cells is halved. For example, out of the 23 pairs of chromosomes in humans, only single chromosomes, that is one member of each pair that is haploid, are passed on to the sex cells. This is essential because when the male and the female gametes fuse during fertilization, the normal double, that is diploid number of chromosomes in the pairs is reacquired. The diploid number, as a rule, is expressed as 2n and the haploid number is represented as n. The stages in meiosis. As per the scope of the syllabus, the stages of meiosis are not required. However, just to know the basic about it, meiosis is completed in two divisions Meiosis 1 and meiosis 2. The first is the reduction division and the second is the mitotic division. So, meiosis is reductional division. Okay. In meiosis, the nucleus divides twice. So, we call it as meiosis 1 and meiosis 2. Now, this is not in the syllabus. Chromosomes condense. Here, 6 chromosomes have been taken as an example, 3 from the male and 3 from the female parent. They have been shown in 2 different colors for the same reason. Then you can see what happens is the chromosomes arrange in homologous pairs. Please remember, homologous chromosomes are the chromosomes which are identical with respect to their size, shape, structure, as well as the genes they carry. They split lengthwise, that is longitudinally. Then you can see that the first reduction division produces 2 haploid cells. These undergo second meiotic division to produce 2 haploid cells each, and as a result in meiosis, one parent cell produces 4 haploid cells. Now, what is the significance of meiosis? In meiosis, the chromosome number is halved, that is, it becomes half in gametes, so that on fertilization, the normal number is restored. It provides for mixing up of genes, very, very important. So it brings about variations. Please remember, mitosis does not result in variations, but meiosis brings about variations. The maternal and the paternal. Maternal is from the mother's side and paternal is from the father's side. The chromosomes, maternal and the paternal chromosomes get mixed up during the first reductional division and they separate on. This is something again important. Cross-joining. While the maternal and the paternal chromosomes are separated, the chromatin material very often gets exchanged between the 2 members of a homologous pair, resulting in genetic recombination. This word is important. Genetic recombination is brought about only in meiosis, not in mitosis. So both these permutations and combinations provide for the innumerable variations in the progeny. That is how the children of the same parents, howsoever similar, are different from each other in certain respects. The variations often contribute in evolution. So in a nutshell, remember, meiosis helps in evolution, whereas mitosis does not help in evolution. And these are the points of differences given in between mitosis and meiosis. Mitosis occurs in the somatic, that is body cells. That is all the body cells other than the reproductive cells. Whereas meiosis occurs in the reproductive cells, that is the germinal cells or germ cells. Second, mitosis occurs to provide for growth and replacement, whereas meiosis helps in gamete formation. Mitosis occurs continuously throughout the life, whereas meiosis occurs only in the reproductively active stage. Mitosis results in the production of 2 daughter cells. Meiosis produces 4 daughter cells. In mitosis, full set of chromosomes is passed on to each daughter cell. This is the diploid number of chromosomes, whereas in meiosis, only half the number of chromosomes is passed on. This is the haploid number of chromosomes. In mitosis, the nucleus divides once, so a single nuclear division occurs after chromosome duplication, whereas in meiosis, 2 nuclear divisions occur after chromosome duplication. Mitosis produces identical cells, meiosis produces randomly assorted cells. Between the gametes are produced and this results in genetic variations. Now friends, just an extra information for you that is about stem cells. Stem cells means undifferentiated cells. That means the cells are similar to each other. They are not differentiated. That is, they do not show any differences among each other. So the term stem cells is very much in the news these days, and especially so in connection with the treatment of certain human diseases, because they have the capacity of unlimited divisions. So they can give rise to one or more different types of cells. The term stem, though more familiar with respect to plants, has a wider sense meaning here, that is branch off. The plant stem bears unspecialized cells that can differentiate into leaves, flowers, fruits, seeds, etc. In animals, there are embryonic cells which differentiate into different tissues and organs. Remember, stem cells are used, they're very, very important. They can be retained and can be used in case of emergency to regenerate a particular tissue or organ. Embryonic stem cells take different development directions. For example, some become nerve stem cells, some become specialized to produce blood cells, and so on. These tissue-specific stem cells persist throughout the life, even in the adults. So friends, with this we have completed the description in details of the chapter cell division. Very soon we will catch up for MCQ solving session. So please stay connected. If you're new to the channel, kindly subscribe to our channel and hit the bell icon so that you will get to know about the next live streaming